So hopefully that's visible for everyone. Yeah, thanks for inviting me and for the opportunity to speak. I'm a PhD student at the University of Toronto under the supervision of Dr. Randy McIntosh. And together we've really been working to better understand early developmental brain networks and their dynamics. Specifically, my work is focused on using a tool known as the virtual brain, or TVB, to model brain network dynamics in early childhood specifically. Okay, so to start, the brain is widely regarded as a complex nonlinear system with neural components that interact both within and between different levels of organization. They also each have their own spatial and temporal scales. And while our measurement tools try to collect or capture collective activity as a whole brain network, so for instance, functional MRI, MEG, or EEG, what we don't really have from those measurement tools alone is a way to understand underlying mechanisms. So what is actually happening at the scale of the cell and molecules, et cetera. And TVB provides us a means to link those different scales and levels of organization that we're interested in. So briefly, what is the virtual brain? It's a neuroinformatics platform that integrates neuroimaging data to create large-scale models of brain dynamics. It can be used to develop personalized brain network models with individuals' own personal brain imaging data. It has potential clinical applications for disorders, for instance, testing interventions and treatment plans, and it's also open source. So if you wanted to download it today, you could try and start using it. As a very brief overview of how it works and the overall workflow, the input for TVB is subject structural neuroimaging data, which kind of acts as the scaffold or the pipes that we work off of. We then specify and select a dynamical systems or neural mass model, which is essentially a mathematical framework that's used to describe the dynamics of local neuronal populations. And then TVB produces simulated network dynamics. So in my case, I'm interested in simulated functional connectivity and functional connectivity dynamics. And importantly, TVB also sets up this empirical validation cycle. So we can use subjects' own empirical functional data in order to optimize their simulations based on particular data features. So trying to get features in our empirical data to be seen within our simulated data. As I mentioned, TVB has potential applications to clinical populations. It's been used in adult populations to identify biophysical parameter values that correlate with numerous clinical phenotypes, including determining predictors of cognitive decline relating to Alzheimer's disease, and identifying biomarkers of motor function recovery in stroke patients, as well as modeling seizure propagation patterns in epilepsy patients. In an aging population as well, TVB has recently been used to identify how global modulation of brain dynamics by structural connectivity, so captured by this global coupling model parameter that we have within TVB, was found to increase with age and SC deterioration. The trend was suggested to contribute to reduced dynamical flexibility and poor cognitive performance. So despite all these findings and use cases in adult populations, we're also really interested in using TVB for modeling and studying early developmental changes. For instance, exploring how this global coupling model parameter is related to brain dynamics and outcomes in children. So that's really our main goal, to evaluate TVB's capacity to model early stages of brain development so we can better understand the brain and its changes across the entire lifespan. Why early development? Early development is characterized by dynamic and rapid changes in many brain features, including the brain's neurophysiological and anatomical features. For instance, we see changes in total brain volume, gray matter, white matter, as well as in the structural and functional connections between brain regions. And such changes are also believed to be related to behavior and cognition. As children enter school age, the new challenges and experiences that they face can help to shape and influence network reorganization and neuronal dynamics. And identifying early biomarkers of outcomes in individuals at risk during childhood could really help to improve prognosis and preventative treatment options, which is, again, a part of why we're really interested in this period. So previous literature has suggested that the maturation of long-range axonal projections and synaptic pruning impact network refinement. These changes are believed to underlie developmental increases in integration, so more long-distance and intermodule connections, and decreasing segregation of structural networks, fewer short-distance and intramodule connections. White matter maturation has also been found to follow along a posterior to anterior progression, so back of the brain to front of the brain. In addition to this posterior-anterior axis, a principal sensory-motor to association axis of hierarchical cortical organization, so spanning from these primary sensory-motor in yellow regions to polysensory association regions in purple, has also been observed. And this axis has been seen across numerous neurobiological features, such as electrophysiology, gene transcription, neuron density, and connectivity structure. Functional brain network studies have also found diverse connectivity profiles and developmental trajectories, including changes in segregation and integration with age. Notably, variable patterns of segregated and integrated functional networks have been proposed to depend on the network's position on the sensory-motor association axis. So during early development, functional hubs are localized in primary sensory and motor areas, and then have been found to transition to include more distributed association areas by middle childhood. By studying these whole brain networks, including these spatial variations in structural and functional features, we can really gain deeper insight into how developmental processes shape diverse functional abilities and identify targets for modifying developmental trajectories. So in addition to an interest in structure and static functional connectivity, a notable area of interest also includes functional connectivity dynamics, or FCD, which refers to the non-stationary fluctuations in FC patterns over time. So using a sliding window approach here, which I will discuss in more detail later, we can compute the variability in FCD, which is essentially a metric that quantifies the switching behavior between low and high activity states, and importantly, has also been associated with information processing and cognitive flexibility. Furthermore, widespread neuronal activity changes across the brain are important components underlying dynamic FC patterns, so it's also important to consider multiple scales of brain network dynamics, which really positions TVB well as a very useful tool for integrating different modalities and levels of organization, offering insight into potential mechanisms linking both structural and functional development. So in terms of my methods, the first step, as I mentioned, is actually processing the neuroimaging data into the appropriate input for TVB. So neuroimaging data that I used included anatomical, functional MRI, and diffusion MRI collected by Dr. Sidney Bray at the University of Calgary. We assessed data from 39 typically developing children between the ages of 4 and 8 and at a 1-year follow-up period. So we have longitudinal data. And an open source preschool data set was also used, which included data analyzed from 23 control children with 60 scans between the ages of 3 to 7, so a slightly younger age group. For the pre-processing, the TVB UK Biobank pipeline was used, which is a new automated and open source multimodal MRI processing pipeline, which was adapted for pediatric population to generate the model inputs required by TVB. The pipeline includes processing of the T1 structural T1 images and user-defined parcellations. Furthermore, processing of the functional images in this pipeline includes automated classification and removal of functional images, processing of functional and removal of noise artifacts using FSL's FIX and study-specific training sets. And then the diffusion subpipe includes probabilistic tractography, performed using FSL's ProbTrack toolbox to generate a matrix of streamlines between all ROIs, resulting in our structural connectivity matrices that are input into TVB. And then importantly, the pipeline outputs detailed HTML QC reports or quality control reports. These QC reports allow for efficient and thorough quality checks of the raw, intermediate, and processed outputs, which is essential for supporting the extensive manual quality control of pediatric data, which I was very pleased with, made my life a lot easier. In terms of the outputs from this pipeline, it included structural and functional connectivity matrices for each subject and the FCD matrices. So for the functional connectivity matrices, These were computed for each subject by calculating the Pearson's correlation coefficient of the bold time series for each pair of regions. For the FCD matrices, they were created using that sliding window approach I mentioned, where we calculate the functional connectivity matrix for each time window centered at time point t, generating a time series of FC matrices. The FCD matrix is essentially a time-by-time matrix, whereby element T1, T2 is the Pearson's correlation of the matrices for time point 1 and time point 2. And then lastly, we have our structural connectivity weights matrix that encapsulates the probability of connection between our ROIs. So now that we have our structural connectivity data that's input into TVB, we also need to specify an appropriate dynamical systems model. So for our work, the Montbriot neural mass model was used to describe the dynamics of each node, or each region of the brain that we're interested in. It describes each node's dynamics by the average firing rate and average membrane potential variables, so R and V. And then we also have this input current I that gives rise to the oscillatory dynamics of a node, where the firing rate of other nodes is weighted by the structural connectivity edges. So looking a little bit closer at our model parameters, we have a number of model parameters with values that are set in accordance to previous studies. And then we also have this global model parameter G, the global coupling scaling factor. That is a modulation index that influences the structural connectivity's impact on each local node's dynamics. Then we also have an external Gaussian noise factor epsilon and its variance sigma. And this noise variable helps to introduce stochastic dynamics into the system. We then use an approach called parameter space exploration, or PSE, where for each subject, independent simulations were created for a range of values of G and noise. And we chose to focus on these model parameters based on several other studies, as well as the critical and potentially predominant role of structural changes in development. So this step is that kind of parameter optimization step where the model parameter values are adjusted iteratively and optimal values are selected based on particular features of interest. So which G and noise values, for instance, give us the best fit between the simulated and empirical data. Looking a little bit more, or a little bit closer at the metrics that we use, for each simulation, we computed 3 different metrics. First, the KS distance between our simulated and empirical FCD matrices. So we wanted to minimize how different the empirical and simulated FCD matrices are. We wanted to maximize the variance of our simulated FCD matrix. So that's the metric that quantifies that switching behavior between low and high activity states. And then we also wanted to identify the model parameter combination that gives us the max correlation between our simulated and empirical FC. So as an example of our parameter space exploration, where we run many simulations with different G and noise values, we can get a plot seen here, where each value corresponds to the correlation between our empirical FC and the respective simulated FC matrices from each simulation that was run. So we see that the simulation that gives us the highest correlation between the empirical and simulated data for this like dark blue band would give us the optimal noise and G values based on that FC correlation metric. Then for each subject, this parameter space can actually be represented as a vector, so we can string the matrix out. And we can also do the same thing for our other 2 model fitting metrics, so for the FCD and the max FCD variance. And that's what we see here. So we have a vector for each of our 3 metrics of interest across each G and noise combination. And to ensure convergence of these parameter spaces based on each of our model fitting metrics, the Spearman's correlations were computed between the 3 respective vectors. And for all subjects, the model fitting metrics parameter spaces were significantly correlated. So then what we did is we took the simulation with the largest combined rank of our 3 model fitting metrics to identify our optimal G and noise values. In addition to that, we also assessed whether the model fitting metrics varied across age using linear mixed effects models. The results revealed no significant associations between age and any of the 3 model fitting metrics, suggesting that our model fits were similar across participants and time points. So we are happy with that. And then next, in order to assess the identifiability and validity of our modeling approach, we also assessed the parameter spaces for a number of different conditions, including fitting to the original simulated data, randomized initial conditions, and fitting to unseen data. really to see if our simulations converge to a consistent parameter space for each individual. So to very briefly, again, summarize where we're at. So what we did for our original parameter space exploration, where we input our structural connectivity matrix, we produced simulations for different G and noise combinations, compared it to our empirical data, and then for each subject, the parameter space exploration is represented as a vector for the combined ranks of our three metrics of interest for each G and noise value combination. So that's what we see here at the top. We have our first original parameter space exploration. And for our second PSE, we ran by doing the whole thing again for a range of G and noise values. This time, rather than fitting the simulations to the empirical data, we actually fit to the new simulated data, to the original optimal simulation. So we take that. The combined rank of our three model fitting metrics was calculated for each model parameter combination, represented again as a single vector for each subject, and then the correlation between the subject's original PSE vector and our second PSE vector was computed to evaluate the model's identifiability. In addition to fitting to the simulated data, we also wanted to evaluate whether the initial conditions impacted the parameter space. So in our simulations, noise seeds are values that are used to initialize a random number generator that controls the pseudo-random pattern of noise in the simulation. So for our original PSE, our seed was set to the default value of 42, and we see in blue that was our original parameter space exploration. We then ran 5 separate parameter space explorations for every single subject with different initial conditions or seed values. So for example, we then set it to 21 and get our combined rank vector. And then again, the correlations between the combined rank vector from the original PSE and each respective seed PSE were computed for each subject. And by using multiple noise seeds, we can really ensure that our results are robust to different sequences of noise. And then lastly, we also evaluated our model by fitting to new data. So for our original parameter space explorations, we actually computed our empirical FC and FCD matrices based on the start of the scan of the empirical bold time series specifically seen here in blue. So we compared our simulated data to the empirical data from the start of the scan. To further validate the models, an additional parameter space for each subject was conducted by fitting the new simulated data to the empirical features calculated on the end of the scan. And then once again, we took the correlations between our original and the parameter space fit to the end of the subject scans and correlated those. Okay, and the results revealed that the subject's original parameter spaces and each subsequent PSE were significantly correlated for all subjects, again, just suggesting that the models were identifiable and reliable. Now that we have established that our modeling approach is robust and we're happy with that, we are interested in exploring the optimal model parameters that we identified. So the G and noise value combination that actually gave us the highest combined rank of our 3 model fitting metrics. So we ran linear mixed effects models with these optimal model parameter values for each subject to assess the associations between these model parameters with age. And the model with G as the dependent variable indicated that G was not associated with age. On the other hand, we found that noise variance was positively associated with age, indicating that older children exhibited greater noise variance. So the findings suggest a generally stable global influence of structural connectivity on the brain networks over the assessed age range and follow-up period, but an increasing level of network variability. So in addition to our optimal model parameter values, from the optimal simulations, we can also assess the simulated local biophysical model parameters, or RNB, so that firing rate and membrane potential, in order to quantify the local dynamics or bistability of each node in the network. So what is this bistability? So here we see for each node or each region that we're interested in, we have a time series for the membrane potential V, as well as the firing rate R. And these two things can be plotted in a phase plot that displays this bistable dynamic, where we have membrane potential on the y-axis, firing rate on the x-axis, and we see we have this upstate focus of high firing rate, positive membrane potential, and then this downstate fixed point. I like to think of it kind of like a coin that you throw into one of those funnels at a science center where you have the coin spiraling at the top of the funnel to an upstate fixed point. So that's the upstate focus. And then the downstate fixed point would be the coin kind of hitting the bottom. But then what you get is kind of a switch in the state where it can return back up to that upstate focus and fixed point. So by simulating these regional bistability dynamics, we can actually ensure the functional switching behavior in our functional connectivity data. That's considered to reflect empirical dynamics and is related to cognition or cognitive flexibility. And from these time series, we can derive a single bistability metric for each region for every subject that essentially quantifies how much a particular region exhibits this bistable behavior. And what we found was the average regional bistability values across all subjects appeared to have a distinct spatial distribution that follows the posterior to anterior or sensory motor association distribution. So we see less bistability in these blue regions that appear more anteriorly or in association regions. So from this, for each subject, we then analyze the spatial distribution of the bistability metric by computing the correlations between the bistability metric across each region of the brain and the posterior anterior axis, specifically the x-axis coordinate of the ROI parcellation. So we essentially get a bistability PA gradient metric for each subject. So a single value for each subject that tells us how much they expressed a posterior anterior distribution of bistability. We also do the same thing for each subject's bistability values for each region and the rank of the regions along the sensory motor association axis. So again, every subject, a single value that tells us how much they express a sensory motor to association distribution of bistability. And then just to summarize what we have, so we have our optimal global coupling and noise variance model parameters. We have our bistability metrics, including the values of how much individuals' regional bistability exhibits those gradients of distribution. And then we related the bistability gradient metrics to both model parameters with linear mixed effects models. And what we found was that global coupling was negatively associated with the expression of the posterior anterior and sensory motor association gradient of bistability, such that subjects with smaller G values exhibited more bistability posteriorly and in sensory motor regions. Or you can think of it the other way, where subjects with larger G values express more bistability anteriorly and in association regions. So overall, we found that noise variance increased with age in early childhood models. This noise variance metric has previously been associated with greater information processing. Therefore, the increase in noise variance with age may reflect a shift in the network's ability to explore the phase plane, leading to a more dynamic network that can better adapt to stimuli or environmental conditions. Previous literature has suggested that brain signal variability or complexity is an essential aspect of brain function that supports better information processing and cognitive flexibility. We also found that the expression of bistability was not uniform across the cortex, but rather followed distinct spatial gradients. The posterior to anterior or sensory motor to association spatial pattern has been observed previously across multiple scales, suggesting that our models are capturing a spatial aspect of brain dynamics established in early development. And furthermore, the extent to which subjects express the spatial distribution of bistability was found to be related to their structural networks level of global coupling, really highlighting the role of brain structure in modulating the dynamics of brain networks in childhood. So very briefly, some additional considerations. The analyses included a relatively balanced representation of girls and boys, but for greater generalizability of the findings, future work should also consider data collected from multiple sites, a larger sample, a wider age range, and longer follow-ups. MEG and EEG would also offer significant benefits for analyzing network state and regional dynamics with respect to the simulated data due to their high temporal resolution. And next steps could also include investigating alternative neural mass models or model parameters to better understand how alternative factors may shape network dynamics. So all in all, this work really showcases how TVB can be a useful tool for developing robust brain network models for better understanding the mechanisms underlying large-scale functional connectivity dynamics in early development. and providing a foundation and a framework to characterize developmental trajectories that can serve as a reference for studying atypical development, as well as changes across the entire lifespan. So from childhood through adulthood and into older age, really hopefully to provide a more complete view of brain development and aging. And then finally, I have some resources for the virtual brain, including documentation, notebooks, and tutorials. We actually ran a TVB study group previously where we walked through some of the foundational theory behind the virtual brain. We go into more detail about what needs to be done with our neuroimaging data before we can actually start creating our TVB models. We also have code for setting up a simple simulation for an example subject, where we provide actual data for you to kind of get started and play around with these simulations. And then we also go over setting up full parameter space exploration, and then how we can look at, extract, and analyze those outputs. So I'll go over some of those documentation, but I will just say thank you first to all of my collaborators and team members, without which this work wouldn't have been possible. But I will stop my slide sharing and just bring up the GitHub repo for our TVB study group that we ran. So we have, hopefully that's visible, each of our sessions where we have both slides, recordings, and resources. We also had homework for these sessions. So again, the theory, processing of the data, setting up our simulations, parameter space explorations, and some analyses. Specifically, this notebook here in our simulation session. This is a useful document if you're interested, in terms of a brief reminder again of the model that we used, kind of viewing and visualizing this bistable behavior within different nodes of interest that you might have. We have some documentation of the packages that are required, as well as visualizing the input for TVB. So that structural connectivity matrix, which you want to look at and ensure is reasonable, like having your interhemispheric connections. And then we also have the actual setup of the simulation. So where we specify the values for different parameters, we select our neural mass model, load in our structural connectivity data, and then actually saving and outputting our firing rate and membrane potential time series. So you can also visualize specific nodes of interest to see that kind of spiking behavior across those variables, and then plotting again that bistable dynamic phase plot, depending on the region of interest you want. And then we also have code where you can then transform that data into the bool time series. So you can visualize the carpet plot and see periods of activity. And then again, also code for kind of creating those FCD matrices, which you can assess as well across individuals. So we have 2 subject examples. So hopefully that's useful. I'm happy to take any questions now.